Good morning and good afternoon, everyone. This is David Piper from Thermo Fisher Scientific. I'd like to welcome you to our keynote talk on genome editing and stem cell engineering for disease modeling. I'd like to thank Dr. John Chestnut for joining us today and to give you a brief introduction of his background. He received his PhD in cell and molecular biology from the University of California, Davis, and followed that with postdoctoral studies at the University of Colorado, looking at recombinant antibody technology. John's had really a broad experience in synthetic biology, I think before it was called that, really. When he started his tenure here at Invitrogen, he really was responsible for developing and productizing the topo and gateway cloning technology that many of you are likely familiar with, and that was a way to shuttle genetic content amongst many different vectors that were suitable for expression in a whole host of organisms. And he went from there to sort of the next logical step of defining targeting strategies using integrases to put material into pre-selected cassettes in various types of cells, but specifically and perhaps of his main interest within stem cells. And that then really was a jumping off point for him to begin leading the synthetic biology efforts and genome engineering efforts at Thermo Fisher Scientific, where he's now responsible for developing an extremely wide variety of tools that allow for the editing of cells, the detection of those edits, and downstream cellular analysis of them. And I think he'll give us a broad perspective of that today. I'd just like to follow up with a couple quick comments before he gets started. One, during the talk, please feel free to submit questions directly to John using the Q&A box located in the bottom left corner of your screen. And again, I'd like to thank you all for joining and invite you to continue your conversation around gene editing following the talk at our stem cell research booth scheduled chat in the exhibit hall.
Okay, thanks, David. Welcome, everyone. It's a pleasure for me to give this keynote, this broad overview of a very exciting field. Today I'm going to just take a high-level view of some of the technology drivers that have really gotten us to this, I'll call it a tipping point of technology that really enables us to do some very exciting things to create new disease models to push forward biological research and drug development. So I'll start with our first slide to really kind of take a broad view of genome editing. Basically, we have our chromosomal DNA, and we design vehicles that will direct certain activities to the chromosome, hopefully specifically and efficiently. The activities, these enzymatic activities or fluorescent activities or labeling activities could be activators, transcriptional activators. They could be transcriptional repressors. They could be labels, as I mentioned. Today what we're going to discuss is specifically delivering nucleases that create breaks in the DNA. So the important part is that these tools will create, in our discussions, will only create double-stranded breaks. And the more efficient and specific we are creating these double-stranded breaks only at the locus that we're interested in, the better off the workflow is downstream. So once we create this double-stranded break in these cells, we can rely on the cell's repair machinery to repair the break, and the mammalian cells tend to be somewhat sloppy about this, so then what generally happens at these repair sites is that there's an insertion or more often a deletion of genetic material at these sites during the repair process. If we create this double-stranded break in a protein coding open reading frame, then this sloppy repair will more often than not result in a frame shift in the reading frame, leading to a premature stop codon and effectively knocking out that gene. If, along with our nuclease, we deliver DNA that has homology to either side of the breaks, we can then replace DNA, so we can either repair single nucleotide polymorphisms, mutations, or we can induce model mutations into the cell line. If we use some larger donor DNA that has homology to both sides of that double-stranded break, now we can add either new elements, including labels, or new genetic coding regions. So really, the crux of the entire genome editing issue or problem is creating this double-stranded break in the genome efficiently and specifically. Application-wise, there's a broad amount of applications that we and others are interested in. Today, we'll focus on cellular disease models. We can create the disease models in animals as well. Cells as factories, so we can modify CHO cells or other cells that are creating biopharmaceuticals to make more robust drugs. As we get better at growing out disease or tissue models in 3 dimensions, we will use what we learn in the cellular models to create the tissue disease models. And then also, there's a lot of work going on in transgenic plants to create disease-resistant and more robust crops, as well as carbon sources for biofuels. And then if we get, as we learn along these other applications, we'll eventually be, as a field, pushing into gene and cell therapy. And I just have one parting shot on that. So generally, we can just study gene function. We can target gene mutation, as I mentioned. We can add transgenes into the genomes of animal models. We can label. We can add stable integration, and eventually tissue and cell engineering for novel functions. So if we look at the cell engineering cycle, as we call it, as a synthetic biology group, we look at it as type of a design, build, and test model. So first, if we have our cells, we want to understand the issues that confront us or what the type of experiments we want to do. One of the main drivers I'll discuss is sequencing. So next-gen sequencing is providing volumes and volumes of data as to what the genetic makeup of particular cell lines might be. And now we can use that data to be able to understand the types of mutation profiles that particular disease models contain. We also can use our in silico tools to take this data and then help us design specific tools that we're going to use to create our edited genomes. Once we understand how to create our tools, we can then use DNA synthesis to create some of the pieces that we want to put in, the donor DNAs. Eventually, as the field matures, we'll be able to put in larger pieces of DNA, including, I think, genomic circuits that we'll be able to assemble from a parts list of genomic parts. So then getting down into the genome editing tools is another of the technology drivers, as you probably know, or I hope to convince you later on in the talk that these tools are becoming much more accessible. We call it democratized. They're very efficient and easy to use. Efficiency does rely oftentimes on efficient delivery of those tools into the cell. And then one of the major pain points here is how do we identify and isolate the properly edited clone. As I hope you'll see, is that increasing the efficiency of the tool, creating the double-stranded break, and then the efficiency of delivery will then make the identification of that final clone less painful. And one of the things that I won't touch today, but I think is an area that warrants specific attention, is how are we analyzing what we've done to our cells, right? So we can sequence the genome, we can do metagenomic and metabolomic analysis of these cells, and generate volumes and volumes of data as to what we've done to our particular cells. and the effect of these edits in particular signaling pathways and other systems within the cell. The hard part, specifically for biologists like myself, is to take all this data, collate it into a model that is easily understandable, something that we can create a hypothesis with. So this is an area that I think you'll see in the future being more developed and being more useful for biologists. So the idea is then, as we run around this cycle, it's a circle until we really get the cell model that we think that we can apply to our particular problem, and then we can then use these modified and validated cells for, in this case, disease modeling or screening for new drugs and so forth. So just to touch base on technology drivers, one of the drivers that I didn't mention, but I think we're all aware of, is now, within the last several years, we can efficiently generate induced pluripotent stem cells from healthy and diseased patient tissue. So we can take cells from a patient, we can create iPS cells, we can then sequence them to understand the mutation profile that might be associated with their particular disease, and then engineer them, and then we can hopefully then, as we get better at differentiating them into terminally differentiated tissues, then use those tissues for modeling. I'll mention a bit about lower costs and increased efficiency of reading and writing DNA, lower costs and increased efficiency of genome editing as well. So basically, this is just a slide to kind of reiterate the amount of progress that's been made since Yamanaka has described his reprogramming factors. Basically, it's become, I think, pretty commonplace to take a skin biopsy or a blood sample and then reprogram those cells into iPSCs. And then we can take these iPSCs and edit those to either induce or repair mutations, and then differentiate those into particular types to do some screening, or eventually, as we get better at therapeutic applications, we might think of using them as therapies as well. The next focus, as I mentioned, was reading and writing DNA. So over the years, the efficiency of creating larger and larger pieces of DNA has gone up. So now we can create very large pieces very efficiently. Even as you know, a couple years ago, entire genomes were being constructed and synthesized. So the chemistries and the technologies for creating synthetic DNA allows us to be able to create constructs, donor constructs and tools without having to do restriction enzyme and PCR cloning, which is very labor intensive in many cases. One of the major things major drivers in here is the cost. So as we increase our efficiency, the cost per base pair for sequencing and synthesis has gone down. So if we look at sequencing, this blue line, the cost of sequencing per base has dropped off significantly in the last several years, where now sequencing the entire genome is reportedly somewhere in the neighborhood of $1,000. So that's becoming very commonplace, which is leading to the generation of large, massive volumes of data, of sequence data, that then we can use to model particular diseases and make hypotheses from. So meanwhile, as you can see from the orange and the yellow line, the cost of short oligo and gene synthesis is dropping off significantly as well. So currently it's about 30 for a double-stranded gene. It's probably between 15 and 30 cents a base pair. There are several entities out there that are working hard, including Thermo, working hard to push that cost down into new ranges. So I think you'll see that the accessibility of synthetic DNA and our ability to design novel DNA constructs in silico and have those synthesized and delivered to our door is going to just get better and better. Finally, we'll focus on editing tools. So we can sequence our cell lines, we can create iPSCs, we can generate the payloads that we want to insert into these cells to engineer the cells. How do we get them into the genome? So really traditional methods back in time, the original, I think, genome editing tools were meganucleases, which were naturally occurring restriction enzymes that we were able to engineer to bind and to relatively specifically cleave DNA. However, these were very difficult to engineer. A few years after that, out came the zinc finger nuclease fusion. So we took zinc finger proteins that bound DNA, fused them with FOC1 nuclease monomers, and now these delivery vehicles, these in the zinc fingers, can then be addressed to particular parts of the DNA that flank our intended cleavage site. The FOC1 nucleases then dimerize and create a double-stranded break. This was a major step forward in the design nuclease production and development. They weren't necessarily the easiest thing to engineer, and they weren't necessarily the most specific thing, but at the time it was actually a huge step forward. Following on from that, in about 2011, 2010, was the TAL effectors. So these are plant pathogen proteins that were identified, transcription activator-like effectors. Again, these are vehicles that we can then fuse nuclease monomers to. These proteins then have this repeat structure that each repeat is responsible for binding one particular base on the DNA. So now we can go back and back engineer these to address them to any DNA strand in the chromosome. And of course, then we can match those up flanking our desired binding sites. So these were much more efficient for engineering and creation than were the zinc fingers, and they were much more specific. And then most recently, the new kind of kid on the block only a few years ago is the CRISPR-Cas9 system. Again, this is a vehicle. The Cas9 protein is a nuclease, but it is nonspecific until it's hybridized, complexed with a single-stranded, what's called a guide RNA. This system is also very effective, and it's actually very easy to design the specificity end, which is this small RNA molecule, and we'll go into more detail about that. So basically then, once you get these molecules, these nucleases, we can then do the same type of operations that I mentioned earlier. We can do a gene knockout and rely on the cell repair machinery to fix our cleavage site, so create an indel, or we can add donor DNA with homology to either side and either add small pieces or large pieces and so forth. So the goal is to try to increase the efficiency of these operations downstream to make isolating the cell clone that we want more easy. So one of the drivers here is the cost of the editing tools. So if you look back in time, back when I was doing my PhD, we were engineering cells by homologous recombination and be able to make one edited cell line or one edited model, animal model, was almost an entire PhD thesis in those days, and the cost was significant. And then as you can see, the era of zinc fingers comes in around 2008, and then you come down to custom TAL effectors in about 2011, 2012. Meanwhile, the cost for each of these is dropping quickly. The specificity and the efficiency and the speed, the ease of manufacture is going up and getting better. And then now we're down into CRISPRs in 2014 to 2015. The cost of the CRISPRs is going down. And I'll say that we're also working, we've got the cost of the custom TAL effectors down in the range of CRISPRs soon as well too. So suffice to say is now the tools are very affordable, and the tools are very accessible to folks that want to do genome editing. So the main star of the show right now is Cas9, CRISPR-Cas9. As I mentioned, there's two parts of this system. So the first is the Cas9 protein. It's a nuclease that is nonspecific. And then when it's complexed with this, what's called a guide RNA, in nature, this guide RNA appears in two pieces, a tracer RNA, which is thought to be important for creating this protein RNA complex, this ribonuclear protein complex. And then the target-specific CRISPR RNA is the other half that then actually describes the specificity or the address of where this nuclease is going to bind and cut. In reality, most times this is a single RNA. So for use for genome editing, we and others use just a fused single RNA, which works well. So basically, when we can complex this guide RNA with our Cas9 protein, it's thought that this complex then binds and tracks down the DNA until it arrives at what's called a PAM sequence. So PAM sequence is a kind of a street sign that indicates where a potential target locus can be for the CRISPR. And then for the case of strep pyogenes Cas9, the PAM sequence is NGG or NAG. So there's quite a bit of sequence space in there for it to work. The complex is thought to then intercalate in at the PAM sequence to create single-stranded character, which then is thought to allow the association of the CRISPR RNA and with the single strand. And then that is thought to lead to a conformational change, which then brings these two nuclease domains of the Cas9 into position to create a double-stranded break in the DNA. And again, it's very specific, and the double-stranded break is very clean in this case. So this is basically the tool that we'll be discussing from here on out. There's several ways to be able to deliver this tool. So we and others, a few years ago when we started to look at Cas9, we were delivering Cas9 in a guide RNA as a plasmid. So we had plasmids that express guide RNA as well as Cas9, and we can engineer in the guide RNA template into the plasmid and deliver them to the cell. This was efficient, but this wasn't optimal. So we worked in the last couple of years to develop several other configurations, notably as Cas9 mRNA along with purified guide RNA. And more recently, our favorite has been working with purified Cas9 protein and purified guide RNA. And this allows us to pre-complex, so make this RNA protein complex outside of the cell in a test tube and then deliver the complex into the cell using electroporation or lipid. And this has led to the most specific and the most active cleavage results that we've found. The last piece that I'll mention, I have a couple slides at the end on this, is using the lentivirus to deliver the Cas9 and the guide RNA. And this will come in very useful for delivering to hard to transfect cells and also for doing some high throughput screening applications, which I'll just touch on at the end of the talk. And again, no matter how you deliver the Cas9, basically you end up with the same types of double-stranded break and the same types of opportunities to engineer your cell that I've mentioned before. So let's think about the workflow a little bit. We have already understood where we want to edit our cell. We need to design and order our tool. We construct and have our tool delivered, or we construct it ourselves. Then we deliver the tool to the cell, and then it takes some time, 5 to 8 days with a normal cell line to grow them out and to validate them. understanding that iPS and ES cells grow a little bit slower. And then it comes down to screening and clonal isolation. So there's a fair amount of pain points here they were easily identified, especially if you were working with them in the lab. So first of all, tool design and ordering. Some of the design concepts aren't that straightforward, especially for folks who are new to the field. For a company, we wanted to make sure that they were easy to order. It wasn't necessarily that easy to order. And then the turnaround time, the TAT turnaround time, tends to be kind of long at the beginning as well. As importantly, the efficiency and the specificity were an issue. As I mentioned, if the low efficiency cloning or editing tools makes it necessary to screen more and more colonies at the end of the day to get that edited clone that you want. And that's really where the pain comes in, I think, in this whole editing workflow, is the manipulation of multiple different cell lines, especially when they're iPS and ES cells, which are notoriously difficult to manipulate. They grow slowly, so the fewer colonies and clones that you have to manipulate to find your edited clone, the better. So some of the things that we worked on a little bit in the last few years to help these pain points were web-based design and ordering tools. Makes it easy to go in and understand the tool you need and how to order them and add them to your cart. And then streamlined guide RNA production. And these are topics that are in some of the other seminars that are available during the 24 hours of stem cells. As far as efficiency and specificity, I've already mentioned Cas9 mRNA and protein. Right now, currently, the protein is our favorite for most applications. The mRNA it comes in a very close second, I think, to that. Cas9 lenti for difficult cells and for screening, I'll show you. We've also worked on genomic cleavage detection kits for validation. So once you have your pool of edited clones, you want to understand how efficiently you've edited your locus to understand how many clones you need to look at to find the one you need. So we call them a GCD kit. It's genomic cleavage detection. So that's a kind of a cornerstone of what's needed in the lab when you're doing these sorts of operations. And finally, it's something that we and many others are working on is reagents to increase or improve efficiencies of homology-directed repair. So this is actually the insertion of a donor DNA into that double-stranded break. So if we want to create an edit, we use this HDR pathway. And so without making a long story longer, there's the two pathways in the cell that are in contrast to each other. We have the non-homologous end joining pathway, which is responsible for fixing the double-stranded break and for, in many cases, creating these indels that we like for doing knockouts, versus the HDR pathway, which is then important for actually repairing that break by inserting a heterologous piece of DNA. So there's a lot of work going on there, and that's kind of the next big pain point as the field moves from doing knockouts to doing more SNP edits. Okay, so talking about just kind of some of the two different tool sets I mentioned. So our Cas9 editing toolbox that we've worked on basically is a Cas9 protein. We have a guide RNA production system that allows the customer themselves to create guide RNAs in less than a day. We are creating guide RNAs in high throughput in 96 well plates. So instead of having to clone the templates into a plasmid and purify the plasmids and doing that, we can go straight to the guide RNA. That, along with our design tool that I mentioned, creates a system. This Cas9 protein guide RNA complex, again, as I mentioned, is very efficient, and we found it's the highest efficiency platform for delivering CRISPR into mammalian cells that have been reported, as far as I know today. Again, that I mentioned, we also are looking at CRISPR knockout screening platforms. So we're looking at lentivirus, and in this case, we're taking advantage of the fact that lentivirus not only does it transduce multiple difficult to transfect cell lines very efficiently, but it also integrates into the genome, and we can take advantage of this as far as creating a platform that the Cas9 CRISPR complex is being generated over time, and which then drives up the efficiency of knocking out a particular gene in that cell. And so then for screening, we want to drive the efficiencies up as close to 100% as possible to get a clean knockout. We're working with pooled and arrayed libraries and gene sets and for doing these type of screenings. And I won't go through all this, but I have a couple slides at the end just as an example of some of the applications you can use for CRISPR for modeling. Again, the workflow, I just want to have this slide out here to kind of exemplify that the genome editing is only a part of the entire workflow. We have our cell types. We first need to understand if we want to edit a gene. The first important thing that we do in our services group is to understand the copy number of that gene. Like doing a karyotype, if you're in a transformed cell line, oftentimes the gene copy number is more than 2. So then it'd be important to know that. We can also do SNP genotyping to understand the mutation profile, the SNP profile of those cells as it relates to a particular disease state. Now we can attack how do we want to do that. If we want to create a SNP, we need to design our payload and synthesize that donor DNA that has that either the repair or that new SNP. Again, it's genome editing tools that we've just been through. Delivery is very, very important. And then detecting or understanding, profiling your edit to ensure that you've done it efficiently. And then in the end, validation can lead to several different ways to validate. And these are just some of the pathway tools that we provide and that we're building on to kind of help with the whole workflow. So finally, I'll just close a little by talking about some potential applications, right? And these won't go into a huge amount of detail. So as I mentioned, iPSCs have really creation of iPSCs is really leading to the ability to do things that we could never do before. So one of them is obviously we generate iPS cells from a patient, either a wild-type patient or a diseased patient. Now we can edit those cells to either repair or add the mutation to those patient-derived iPS cells. So now we have a matched set of iPS cells, and considering the patient background is identical for both these cell types, the only difference is that particular mutation that we've either fixed or the one that we've induced. And so now we can have a good comparison between these two cell types that we're more confident that any differences we see is only due to that particular mutation and not differences in patient or cellular background. Now, as we get good at differentiating these cells, we can take these down into particular lineage and then be able to do screening or pharmacology or other types of phenotypic or biological study on these cells. So to exemplify this is we did a collaboration with the Parkinson's Institute a couple of years ago. In this case, we took some biopsy samples from several different patients with different types of Parkinson's syndromes. And these are fibroblasts. We reprogrammed these into iPS cells. At that time, we were using tau effectors, and so what we did is we took those iPSCs and we understood and confirmed what the mutation profile in these particular cells were. Then we either induced the mutation or we repaired mutations in these, and then we validated those by sequencing, and then we created neural stem cells from those, and then we wanted to look at and it's ongoing, but looking at the different pharmacology and functional analysis. A little snippet from that study is in this slide. So basically we have a first glimpse in neural stem cells from this experiment. So in this case, we've edited edited the cell lines at the LRRK2 gene, and so then the first experiment on the left is showing that we have all the same number of cells in the wells, and so in the middle, when we look at glucose sensitivity, so in these cells that were the LRRK2 mutations were sensitive to glucose. As we edited those cells with the tau effectors down, now we've reversed that glucose sensitivity to those in those cells, showing the expected result. And then as to confirm that, we took those same cells and then we used a drug in glucose inhibitor to show that we can then now rescue that phenotype from the tau edit, show that we reversed the glucose sensitivity. So basically what we've done here, and this is very, very early snapshot, is that we have created a matched set of disease and cell line iPS cells from patient samples, and very first pass we're seeing the type of pharmacology or biochemistry that we would expect from that genotype. So these cells are still being worked on very heavily as far as understanding the actual differences that these mutations cause, and hopefully eventually for doing some drug screening. A very early example of using edited iPS cells from patients to do interesting disease modeling. The last piece I want to talk about is then using genome editing for functional genomics. And then my examples here are not in iPS cells, but I think that you might agree that we could then use iPS cells in a similar type of procedure going forward. So currently our functional genomics tools are basically we can do gene knockdown with microRNAs and siRNAs. Now recently we can use CRISPRs and tau effectors for doing these similar types of operations and to increase specificity of these things and the efficiency. So now one of the exciting areas that we're addressing is that instead of using RNAi to do gene knockdown, we're looking at using CRISPR to do gene knockouts in libraries. And the thought is that the CRISPR knockouts has the potential for being much more specific than siRNA knockdown. So we're in the process of testing these, and I want to show you just a little bit of data that we're generating to show that these libraries are functioning as expected. So the workflow basically looks like this. We have arrayed libraries of CRISPRs that in each well contains a set of CRISPRs that attack one particular gene, so that will knock out one particular gene. So each well relates to one gene knockout. So the workflow, we would basically have our cell line. We would then test the growth kinetics and antibody killing curves and standard kind of screening procedures. Then we would apply our CRISPR arrays to this set of cells and then do specific assays. We can look at viability. Did that knockout of that particular gene, was that toxic, and did those cells die? That would be very easy to find out. Apoptosis, there's lots of screens for apoptosis. We can do positive screens, so we can look at expression of specific markers or reporter assays, morphologic changes, or high content screening. So there's many, many ways to do that, and by doing this in an arrayed format, it allows us to quickly identify what genes might lead to a particular phenotype. Now, if we take the winners or losers, if you will, from that experiment, then we can follow that up rather easily by looking specific cleavage assays to understand that we did knock out the particular locus that we're looking at. We can use qPCR, RT-PCR around those loci or other associated loci, a Western blot to look at a particular protein level, and also, of course, sequencing to understand what the types of indels that we induced in those particular cells to verify that the phenotype that we're seeing is at least associated with that knockout. So one experiment that we did to start using this system to really see how well it performed is looking at a cell sensor cell line, and this is basically a cell line that has an engineered NF-kappa B promoter that will express the beta-lactamase gene, and this is an ME180 cell line. So NF-kappa B is a signaling pathway responsive promoter, and we'll go into that. So the assay basically looks like we have our cell sensor cell line. We then transduce that cell line with both a Cas9 lentiviral particle along with our arrayed guide RNA lentiviral particles. So it's a two-particle system. Now these then integrate into the genome and allows us to do antibiotic selection over about a week to kind of amplify that cleavage, that knockout. These cells were then transferred to a 384 well plate, and then we stimulated that NF-kappa B signaling pathway with TNF, tumor necrosis factor, and now we can measure using our report out. So the system looks something like this. On the bottom here, you'll see this is our reporter molecule. It's a FRET-based fluorescence molecule. So in the intact molecule, upon excitation, it fluoresces green. And then upon activation of this NF-kappa B promoter, we get expression of this beta-lactamase reporter. The beta-lactamase then comes down and cleaves this reporter molecule at a beta-lactam ring, which splits the reporter molecule, relieves this FRET reaction, and now these cells that have the active beta-lactamase or active NF-kappa B promoter, now they fluoresce blue. So the easiest to remember is that if the pathway is inactive, the cells are green. If the pathway is activated, we get NF-kappa B NF-kappa B expression, and the cells are blue. So upon stimulation of these cells with TNF-alpha, binds to the TNF receptor and then activates this signaling pathway cascade, which then leads to activation of the cells. So one thing we did first is we looked at the cells, obviously. So without TNF-alpha, the cells are green as expected. So the pathway is inactive. Once we stimulate the cells with TNF-alpha, or control cells, the unedited cells, or a CRISPR that is directed toward an HPRT gene, which is not associated with the signaling pathway, these cells are blue as expected. Now, when we targeting in the wells that we're targeting other particular molecules or other particular signaling molecules like IKK alpha, TRAD, and the TNF receptors, you can see here, now we're seeing some disruption of the signaling pathway. And then if we dose in the TNF, as you can see at the bottom, now we're starting to see some pharmacology exist here. So this is an example of using these types of CRISPR knockout libraries to get a specific response in a particular pathway. This is, again, is done in a transformed cell line. I think that doing these in differentiated iPS reporter lines is something that we'll be seeing in the future. So finally, I'll leave you with this. So we talked about disease modeling, we talked about screening. I think all the things that we're doing in these areas are teaching us more and more how to apply these things into potential therapy. Again, we can do ex vivo therapy. These things are occurring now. There's been some great stories about using tau effectors recently for creating CAR T cells and curing some leukemias. And then eventually in vivo, by injecting some of these tools, I expect that these will be useful for doing other types of therapy. So I am going to end with that, and I think hopefully I gave you a kind of a broad overview of the field as I see it anyway, and then and hopefully you'll have some good questions for me. I'll be happy to try to answer them. Thank you.
Well, thanks again, John. That was great. Like a couple decades worth of science. So we do have some questions on the line already, and I would just like to remind everyone, thank you for your attention, and remind you that you can ask John questions through the Q&A box on the lower right. I may have said lower left when we started, but I'm sure you were able to navigate it. So I'm going to go ahead and I think in no terribly particular order here, get through the questions with you, John. One of the first ones that came through was, how difficult is it to engineer heterochromatic loci with CRISPRs, or for that matter, any tool?
I think that the nature of heterochromatic loci is that it's found in nucleosomes and thought to be inaccessible physically to anything, any protein. I believe between the binding of that region to other proteins like nucleosomes and methylation and other sorts of epigenetic remodeling, it's going to be a challenge. It'll be a challenge to address these sites no matter what tool you use. I think there will be more work, I'm sure, on using maybe some epigenetic tricks to open up particular loci to give them access to tools for editing, but that's going to require a significant amount of work.
Thanks, John. Another question was, what is the best method to assess the effectiveness of gene knockdown via CRISPRs?
Well, ideally, the most effective is using an antibody to the resulting protein. If one does not have an antibody to the protein, then you're left with sequencing the locus to understand that you've induced a stop codon to that gene.
Kind of along the same lines there, we've got a listener that had joined a webinar about a month ago that IDT put on, and during that the presenter suggested that they did not have a way of assessing off-target homologous recombination. Could you maybe comment on how you might suggest users look at off-target homologous recombination?
Off-target homologous recombination. That's a tough one. I think there's some tools that are evolving to identify off-target indel formation. I think off-target homologous recombination will be more difficult because what you're looking for might be a little smaller, a smaller target. I think might be able to take some of these platforms such as GuideSeq or BLAST to try to adapt them to identify your donor DNA if it's inserted into the genome, but I think that's going to be very challenging.
Are there tools, so that you mentioned GuideSeq, and that would kind of, I guess, imply looking at it in an unbiased way, but what about, looking at kind of where the light's on, where you might expect it to be based on your designs?
Yeah, I think we can, and what we do currently, we and others, is we can use our design tools and what we understand about CRISPR specificity and PAM sequences to predict potential off-target sites. And we do things like targeted sequencing of these different sites to understand where, if at all, we're getting off-target cleavage. And these could be used, I think, for HR or for homologous recombination off-target. There's ways to do things. I mean, we can sequence the exomes. We can do AmpliSeq, which I mentioned about targeted sequencing. Again, it generates a lot of data, but you don't know what you've missed. Right? So it's like I say, and you brought it up, David, it's like looking for your car keys under the light post. We really would try to develop and find something that's non-biased that we could then actually not miss it.
Thanks, John. This one probably has maybe more than one answer to it, and I think when I look at the workflow slide that you presented, but this listener wanted to know how long will it take to get to the gene editing result. So I guess that may depend on what the result is.
It depends a lot on the result. It depends on things like how fast do the cells double. It will depend on how efficient logistically, how efficient your cleavage is. So if it takes longer to screen, 500 clones than it takes to screen 10 clones, just hands-on wise. A lot of it, I think, is cell doubling time. So cells that grow quickly, you can get to a population of cells that you can analyze quicker. As we know, ES and iPS cells that tend to grow quickly are generally not the ones you want. so it's a bit of a tough situation.
I guess that goes to maybe another listener question here that I'll state specifically the way they wrote it, but generalize it. And they want to know, how easy is it to edit T cells using CRISPR-Cas9? And I guess maybe you kind of generalize that to, for cells that are difficult, what might be the approach that you suggest based on the tool sets that you covered?
Yeah, T cells, that's very popular and very tough cell line to manipulate. So they don't transfect very well. So it's all about, I think, delivery there. As you can see, there's some data that came out from this company Selectus, where they're using tau effectors to engineer T cells quite effectively. But they're engineering healthy T cells to create a bank of cells that they can use as a therapy. By doing it patient-specific T cells, then it's a little bit different, because you have to do it with the cells that you get from the patient, and you generally will have a lower number of cells. I think my broad answer is probably not that helpful. Is they're very difficult cells. It's difficult to manipulate. I don't think they grow very terribly well. You don't have that many doublings. Depending on your population, you might have a mixed population. They might differentiate. So it's a huge challenge. And as we know, T-cell immunotherapy, CAR T cells is a huge area, and is growing fast. And certainly have some folks here in our group that are paying a lot of attention to that field right now.
I think that maybe leads into another question from another listener, which was, what are the latest applications for CRISPR genome editing in gene therapy for specific diseases? So maybe just kind of touched on one.
Yeah, I think so. And I think it's very, very early for CRISPR. I think the gene therapy that we're seeing coming out now in the clinic has been done with tau effectors, which had a little bit of a head start than CRISPR. There are several groups, there are several therapeutic companies that are focusing on cell therapy, specifically with CRISPR. I haven't seen a lot of data come out of them, but that doesn't mean that nothing's happening. So I think it's really early days for gene and cell therapy with CRISPR, but I would be very surprised if we don't start seeing a significant amount of literature in the future.
So that, I guess, ties into another one here. I'll segue. You mentioned that tau's had a head start. One of the listeners wants to know what you think the advantages of CRISPRs over talons might be, or I suppose vice versa, if there's any comment that you might make there.
Yeah, so that's a good one. I think we're wrestling with that too. They both do the same thing, right? They both create a double-stranded break in the genome. As I mentioned, tau can be made rather rapidly and cost-effectively. The challenge with tau effectors is kind of the ease of design, in the sense that You can create, for CRISPR, you can create a new nuclease just by changing your guide RNA. And what I didn't mention is that being able to create these complexes, the ribonucleoprotein complexes outside of the cell, allows you to mix them and you can multiplex. And so we can knock out probably easily 4 genes in one transfection. It's difficult or maybe impossible to do that with tau effectors currently. So you can do multiplexing. On the other hand, Cas9 CRISPR, the specificity is in question. So there's a lot of discussion about off-target. There's some recent literature that seems to address some of that. But the thought now, at least, is that the tau effectors, given their binding specificity and binding track, would be thought to have higher specificity. But there hasn't been a really good survey yet between CRISPRs and taus as far as who really wins on specificity.
Sort of adding on to that, John, I guess one thing that comes to mind for me, I think you touched on this a little bit, is it's partly specificity, but it's the ability to maybe target the taus to more specific locations because they don't have as much constraint around the design right now.
That's a really good point. And so there is constraint for CRISPR as far as you need to have a PAM sequence around where you want to make your cut. And what we do know is that when we're doing editing, inserting a piece of DNA, we want to have that cut as close to the edit as possible. Further away that cut is from our intended edit, the efficiency drops way down. So CRISPR is relying on a NGG or NAG to be around where we want to have our cut. Tau effectors, we have a mutation of tau effectors that allows us to slide that tau complex anywhere on the genome we want, base by base. So we can put that cut directly on top of where we want to create our mutation. So, and then, in theory, then that would give us our best efficiency for gene insertion.
Sort of related to that, we've got, I guess, another listener question about the different kinds of Cas9 proteins that they've read about in the literature, and they're curious as to which one might be the best to use for genome editing applications, and maybe sort of dovetails with what you were just saying.
Right. So there's been some literature out recently, identification of new species of Cas9, and it remains unclear whether the new ones have much of an advantage over the strep pyogenes. There's I think around 30 different species of Cas9 that's been identified so far total, and then there's been a handful that have been identified as being somewhat functional. And then even more recently, there was a paper from Feng Zhang's lab describing some mutations they've done to the strep pyogenes Cas9 that tend to add, that are thought to add specificity. Bottom line, the workhorse that we're still working with is Cas9, the strep pyogenes Cas9. With our protein and guide complexes, we've been able to get very, very high efficiency cleavage at loci in iPS cells in the neighborhood or upwards of 70 to 80% efficiency at some loci. I think for a lot of things that's going to be plenty. And as long as the specificity is good and then there's low or no off targets, I think that's going to be a great tool for quite some time.
Okay. Well, John, thanks. I think we're about running out of time. We did have one other question, which was, can you briefly specify which diseases gene editing has been successfully applied? I'm not sure if they're asking about, disease models or cell therapy.
That's a great question. Certainly the example from Parkinson's is a good one. And the other kind of edited therapy that we've seen is this example of CAR T cells in leukemia. and that's a killer. I mean, it's a great example. Sorry. So I think that as far as other examples of particular screening, I'm not that aware, but actually doesn't mean they're not out there. I think a good question. It's going to spur me to go back and look in the literature to make sure I'm up to date.
Okay, and we have this one last one. We're about over the limit, but we had sort of more of a technical question, which is, I don't think we can give you a real answer, but is there any limit in length of the donor insert for CRISPR, Cas9, or TAL systems?
Great question. What we're seeing is that as the donor insert gets larger, the efficiency drops off. So I think that we've seen up to a couple KB insertions using TALs and CRISPRs, and even higher than that. I think the key is going to be, are you willing and able to screen enough colonies to get to the right one? So I would say I'd be nervous over maybe 2 to 3 KB, but it depends on your cell background, depends on how efficiently you create that double-stranded break.
Terrific. Well, John, thanks for such a great overview. It's just a huge amount of work that you and others have put in over the last 5 to 10 years in this area, and appreciate it. And I'd like to thank the listeners for joining us today. And really a great set of questions. I hope we've been able to help you out with some answers. Again, I encourage you after the talk to join us in the exhibit hall. We've got R&D experts standing by for a scheduled chat to talk about gene editing, and hopefully they can answer anything that may have crossed your mind that we didn't get to during this presentation. So John, thanks again, and I think we're ready to sign off.
Yeah, I want to say thank you very much. It was a pleasure.